Welcome to BioBits and Bytes. Today we're diving deep into, well, a really fascinating world, how living cells, specifically yeast, so these little industrial workhorses, manage the demanding task of producing valuable proteins, stuff for medicine, for industry. Our listener, the learner, shared some great research exploring the hidden costs, and the clever strategies involved when we turn these single cells into tiny protein factories. So we're going to unpack all of that, explain the key science terms, maybe reveal some surprising things along the way. And with us today is an expert to guide us through this pretty complex topic. Okay, let's get into it. Maybe start with the basics. What exactly is heterologous protein production in yeast? And why is it such a big deal? The learner pointed us to a source, burden imposed by heterologous protein production. Could you maybe give us a clear definition for everyone listening?
Certainly, yeah. Heterologous protein production. Um, in essence, it's the process where we engineer a host organism, in this case, yeast like Saccharomyces cerevisiae or Comagene alfalfae, which you might know as Pichia pastoris.
Right, the common lab yeasts.
Exactly. We engineer them to produce a protein that they don't normally make. It's foreign to them. That's the heterologous part,
Okay, so heterologous just means foreign, basically.
pretty much. And this is, well, it's a cornerstone of modern biotech
Mm-hmm.
because these proteins can be incredibly valuable.
Like what kind of things?
Life-saving drugs like insulin for diabetes or tons of enzymes used in everything from making food to biofuels, even vaccine components.
Wow, okay.
And yeast are really, really good hosts for this. They're relatively easy to grow, easy to genetically modify, so they become these efficient little cell factories for making what we need.
Cell factories, I like that term.
Yeah, and that source, burden imposed by heterologous protein production, it rightly calls this a billion-dollar global industry that really reflects the scale, the economic importance of biopharma and industrial enzymes made this way.
A billion-dollar industry, powered by these tiny organisms. That really highlights why we need to optimize them, right?
Yeah.
Which brings us, I guess, to the main challenge in that same article. Making these foreign proteins, it puts a huge strain on the cell's resources.
That's the core issue, yeah.
Resources like energy, building blocks, stuff the yeast normally uses for its own growth and just staying alive. Could you maybe elaborate on this production burden? What does it really mean for the cell?
It's a critical point. You're asking the yeast to make something that gives it no direct benefit for survival. So it creates this direct competition for the cell's limited internal resources.
Like fighting over energy.
Exactly. And this production burden, it's not just theoretical. You see it happen. The engineered yeast often grows slower. It triggers various stress responses inside the cell.
Mm-hmm.
And ultimately, you often get lower overall yields of the protein you actually want. The cell is basically diverting effort from its own needs to make our product.
You mentioned the term cell factories again. It really sounds like we're treating these living things like mini industrial plants. What does that really imply about how we're using them?
Well, it's a pretty good analogy, actually. We essentially reprogram them genetically. We put in the genetic blueprint, the gene for the protein we want.
Right, the instructions.
Yeah. Then we give them nutrients, keep them in carefully controlled conditions, and as they grow and do their normal thing, they also read our foreign instructions and start making that protein. So yeah, like a factory takes raw materials and energy to make a product, these cell factories use nutrients and their own complex machinery to synthesize our target protein.
But the article also points out something interesting. Yeast have their own ways to, downregulate protein production if it gets too demanding for them. Why is that a problem for us if we want high production? Seems like the yeast is just trying to be efficient for itself.
Exactly. That's the conflict. Yeast have these really finely tuned regulatory networks. They need to maintain balance, respond to stress. If they sense this huge drain on resources for a protein that doesn't help them survive,
Our protein.
right, our heterologous protein, they might see it as a stress, an imbalance. So to save energy and resources for their own essential stuff, they can actually activate pathways that reduce the production of that foreign protein. It's crucial for the yeast's survival, but it directly fights our goal, which is maximizing the yield of that valuable protein.
Okay, so it's a constant push and pull. So what are the specific things causing this burden? Both the burden article and another one, cell factory engineering, seem to point to a few key areas, starting with the foreign DNA itself. Just having that extra DNA around is a problem.
Yes, definitely. The DNA itself contributes in several ways. First, just maintaining that extra DNA, whether it's on
Wait,
plasmids.
hold on. Plasmids. Could you define that for us?
Okay.
And why do they often have these selection markers, like antibiotic resistance genes?
Sure. So a plasmid is basically a small, usually circular piece of DNA that can replicate on its own inside the yeast cell, separate from the main chromosomes.
Extra DNA floating around.
Kind of, yeah.
Yeah.
And we often put our gene of interest onto one of these plasmids to get it into the yeast. Now, the problem is the yeast doesn't naturally want to keep this extra plasmid. It takes energy to copy it every time the cell divides.
Right.
So, to force the yeast to keep it, we add a selection marker gene onto the same plasmid.
Ah.
Often that's a gene for antibiotic resistance.
Ah, I see.
Then, we grow the yeast in media that contains that specific antibiotic. Only the cells that have kept the plasmid and therefore the resistance gene can survive and grow. It lets us select for the cells making our protein.
Clever, but
But, as the burden source points out, making that resistance protein also costs the cell energy and resources. So it just adds to the overall burden.
so the yeast pays a price just to survive in our lab conditions, on top of making our protein. Wow. The article also had a really fascinating point about different types of yeast. It said diploid S. cerevisiae cells with 2 chromosome sets might actually lose plasmids under burden, but haploid strains with just 1 set tend to keep them. What does that tell us about how they cope with stress differently?
That is a really insightful observation. It suggests that diploid yeast, maybe because they have more genetic backup, might prioritize their own growth when things get tough. They might just ditch the burdensome plasmids, stop making our protein and the resistance marker to save energy.
Giving them a competitive advantage, maybe?
Potentially, yeah. While haploid cells, maybe with a different resource strategy, might hang on to the plasmid, perhaps keeping it, even at a lower production rate, is a better bet for them in that stressful situation. It really shows how the basic biology of the host cell, like its ploidy, can really affect how stable and productive our system is.
Which makes total sense why the article then mentions integrating the gene directly into the yeast's own chromosome without needing selection markers as a better strategy to reduce burden.
Exactly. If the gene is part of the main chromosome, it gets copied automatically when the cell divides. No need for plasmids, no need for constant antibiotic selection pressure, and often much more stable expression over time. It generally leads to less stressed, more reliable cell lines for large-scale production.
Okay, shifting gears a bit. Another big factor mentioned is the number of copies of our the gene dosage. The article talks about a Goldilocks range. Not too few, not enough protein, but also not too many because that overloads the cell.
Yes, that's a crucial concept.
It even gave examples in K-Fafie for lipase, xylene, insulin, where more gene copies didn't always mean more protein. That's kind of counterintuitive, isn't it? Why doesn't more DNA always equal more protein?
It seems logical, right? More blueprints should mean more product. But the cell's machinery and resources are finite. If you have too many copies of that foreign gene,
Yeah.
the machinery that reads the DNA transcription and makes the protein translation, especially the ribosomes, they just get overwhelmed. It creates a bottleneck.
Like a traffic jam inside the cell.
Exactly. And it drains the pool of essential building blocks, amino acids, energy like ATP, those redox cofactors like NADPH we need for making amino acids. All things the cell needs for itself, too.
Oh, okay.
So push the gene dosage too high, and the burden becomes so great that the cell actually gets less efficient at making the protein. It might even trigger stress responses that shut things down. That Goldilocks range is the sweet spot. Enough gene copies for good production, but not so many that you cripple the cell.
That balancing act again, it really highlights the complexity, and the cell factory engineering source mentions these modern genome editing tools like CRISPR-Cas9, maybe fused with piggyback, and prime editing. How do these help? Are they letting us get more precise with that gene dosage and location?
Oh, absolutely. These are game changers.
Oh.
Older methods of getting DNA into the genome were often random. You didn't know where it would end up. It might disrupt other important genes, and controlling the number of copies was really hard.
Leading to lots of variation.
Huge variation. But tools like CRISPR-Cas9 and prime editing let us target our gene to specific known locations in the yeast genome, places we know are good for stable, high expression, and crucially, they give us much better control over inserting a specific number of copies.
So more predictable result.
Much more. It minimizes disruption to the host, leads to more consistent production, less variability between cells. It's key for building those robust, reliable cell factories we want.
Okay, so that's the DNA burden. Let's move to the next source. Making the protein itself translation. The burden article stresses this needs tons of amino acids, energy, ATP, and those NADPH cofactors you mentioned, which are needed for making amino acids.
Absolutely. Protein synthesis is one of the most energy-intensive processes in the cell. Every single amino acid needs energy to be activated and linked into the chain. When we force the yeast to make massive amounts of our protein,
Yeah.
the demand for those basic building blocks in energy just skyrockets. It can really deplete the reserves the yeast needs for its own essential proteins and other pathways.
And NADPH seems really critical here for making the amino acids in the first place. The source talked about different ways yeast make NADPH, the pentose phosphate pathway,
Right.
DQP and K-FAFI, and acetate formation in S. cerevisiae. And maybe boosting these could help. Can you explain those pathways briefly and why something that works in one yeast might not work in the other?
Sure. So the pentose phosphate pathway, or PPP, runs alongside glycolysis, the main sugar breakdown route.
Mhm.
A key job of the PPP is making NADPH, which is vital for building things, like amino acids.
Okay.
Acetate formation is another metabolic route S. cerevisiae uses to make NADPH, especially under certain conditions. Now, why might boosting PPP work in K-FAFI, but maybe not S. cerevisiae? It comes down to differences in their core metabolism, how they regulate molecule flow.
They're wired differently.
Essentially, yes. The source notes that in K-FAFI, when it's burden making our protein, the PPP often naturally ramps up anyway to meet the NADPH demand. So engineering it to work even harder can help.
Makes sense.
But in S. cerevisiae, under similar stress, the PPP might actually get downregulated, and maybe acetate formation becomes more important for NADPH. So just blindly trying to boost the PPP in S. cerevisiae might not work, or might even be counterproductive. You really need to tailor the metabolic engineering strategy to the specific yeast you're using.
That's a great point. Need those species-specific strategies. Now, the cell factory engineering source brought up something kind of counterintuitive. Just the act of overexpressing our protein can itself burden the translation machinery. And that could trigger stress responses that, ironically, shut down our gene.
Yes, that's a really important feedback loop to consider.
Yeah.
The cell's capacity to make proteins isn't infinite. If you push it too hard, demanding huge amounts of one specific protein, you can overwhelm the ribosomes, the protein factories,
Right.
leading to backups, maybe misfolded proteins. This congestion can trigger stress responses aimed at restoring balance. And one way to restore balance is to simply stop making the thing that's causing the problem, our heterologous protein.
So you shoot yourself in the foot, basically.
Potentially, yes.
So how do we deal with that? Cell factory engineering mentioned using non-coding RNAs, microRNAs, long non-coding RNAs, LNC-RNAs, to sort of fine-tune translation. How do these RNAs work? And they also mentioned maybe using miRNAs to dial down apoptosis, program cell death. Why would that help?
Right, non-coding RNAs are fascinating regulatory molecules. They don't code for proteins themselves, but they control other genes. MicroRNAs, these tiny RNAs, can bind to messenger RNAs and either block them from being translated or cause them to be degraded. Less protein gets made from that target gene.
Okay, like putting the brakes on slightly.
Exactly. Long non-coding RNAs are more diverse, longer, and can regulate genes in lots of ways. The idea here is, instead of just pushing for maximum protein production all the time, which risks triggering those shutdown responses, maybe we can use these RNAs to achieve a more balanced, sustainable level of production, fine-tuning it.
And the apoptosis part, dialing down cell death.
Well, the intense stress from making tons of foreign protein can actually push the cell towards initiating programmed cell death, apoptosis.
Yeah.
If we could use something like miRNAs to gently dampen that self-destruct signal,
Yeah.
the cell might tolerate the burden longer, survive better, and ultimately maybe produce more of our protein over the entire production run. It's about keeping the factory running, even if it's under stress.
Interesting strategy. Okay, so the protein is made. What about getting it processed and secreted? The sources highlight this journey, starting with getting into the endoplasmic reticulum, the ER, as another potential bottleneck, the ER being that internal membrane network for protein folding.
Exactly. For many valuable proteins, especially things like antibodies, we need them secreted out of the cell to collect them easily. This involves the protein entering the ER lumen, folding there, maybe getting modified, then moving through the Golgi apparatus for more processing, and finally getting packaged into vesicles that release it outside.
Sounds complicated.
It is. Each step needs specific machinery, energy, and any step can become rate limiting, adding to the burden.
The Burden article mentioned the signal peptide, that little tag that directs the protein to the ER, is critical. And it noted that getting the protein into the ER as it's being made, co-translational, might be less burdensome than making the whole thing first and then importing it, post-translational. Why would the timing matter so much?
It likely matters because co-translational entry gets the protein chain into the protected folding environment of the ER right away. Folding can start immediately, potentially preventing misfolding or clumping up in the crowded cytoplasm outside the ER.
Misfolding is bad, obviously.
Very bad. It triggers stress, needs energy to fix or degrade. Post-translational import, where the whole protein is made first, might put more strain on the ER's import machinery,
Right.
especially for large or tricky proteins. It has more chance to misfold in the cytoplasm before it even gets to the ER.
And the article mentioned that even a common signal peptide, the K-FAFI alpha factor 1, can sometimes increase burden.
Yeah, that suggests it might not be optimally efficient for guiding every protein across the ER membrane post-translationally. Maybe it leads to delays, more chaperone interaction, higher energy costs for certain proteins.
And cell factory engineering adds that if that signal peptide isn't cut off correctly, miscleavage, you get junk protein. It mentions engineering signal peptides to work better with the SRP, the signal recognition particle. What's the SRP and what does this tell us about secretion complexity?
Okay, so the signal recognition particle, or SRP, is this complex that grabs onto the signal peptide as it emerges from the ribosome. It pauses translation and drags the whole ribosome mRNA protein complex to the ER membrane, helping the protein enter.
A delivery service.
Kinda, yeah. Efficient interaction between the signal peptide and SRP is crucial for getting things started correctly. If the peptide isn't recognized well, or if it gets cleaved at the wrong spot later, the protein might end up with the wrong structure, won't fold right, won't work.
So tweaking the signal peptide matters.
Hugely. Engineering them to bind SRP better can improve secretion. But the fact that the best signal peptide varies depending on the protein you're making and even the specific yeast strain, it just shows how incredibly complex and non-universal this whole secretion process is. It often needs optimizing case by case.
Okay. Inside the ER, proper folding is key, especially for proteins with disulfide bonds, those molecular staples holding the structure together. The Burden article says overloading this can cause oxidative stress involving glutathione and reactive oxygen species, ROS. Can you unpack that? What are disulfide bonds, glutathione, ROS, and how do they link to stress?
Sure. Disulfide bonds are strong chemical links between sulfur atoms on cysteine amino acids in a protein. They're vital for stabilizing the 3D shape of many proteins, especially secreted ones that face the outside world.
Okay, like internal scaffolding.
Good analogy. Now, making these bonds happens mainly in the ER, and it's a redox process involving electron transfer. If you overload the ER trying to fold tons of protein with lots of these bonds, you can disrupt the ER's delicate redox balance. This can lead to more reactive oxygen species, ROS, being generated as byproducts. ROS are damaging molecules that can harm proteins, lipids, DNA.
Oxidative stress.
Exactly. And glutathione is a key molecule the cell uses to normally neutralize ROS and maintain that redox balance. But if ROS production overwhelms the glutathione system, you get oxidative stress, which adds significantly to the cell's burden.
The source mentioned maybe overexpressing things like glutathione peroxidase to detoxify ROS or HAP1 to boost the stress response, so we can try to help the cell cope.
Yes, those are potential strategies. Glutathione peroxidase directly helps neutralize ROS. HAP1 is a regulator that turns on genes involved in responding to oxidative stress. Boosting these could enhance the cell's own defenses, making it more tolerant to the stress from high protein production.
Now, several sources mention the unfolded protein response, the UPR. This sounds like a big deal. It's a defense mechanism when the ER gets overloaded with unfolded proteins, right? The IR1-HAC1 pathway in F. cerevisiae was given as an example. How does the UPR try to help, and why might it sometimes not be enough or even be counterproductive?
The UPR is absolutely critical. It's how the cell senses that buildup of unfolded proteins in the ER, a classic sign of burden from heterologous production. The IR1-HAC1 pathway is a key part of this in yeast.
How does it work?
IR1 is a sensor in the ER membrane. When it detects unfolded proteins, it gets activated and performs a unique splicing reaction on the messenger RNA for a transcription factor called HAC1.
Splicing the mRNA. Okay.
Yeah, this processed HAC1 mRNA then gets translated into the active HAC1 protein. HAC1 goes to the nucleus and switches on a whole set of genes designed to fix the ER problem.
Like calling in reinforcement.
Exactly. Things like molecular chaperones to help fold proteins, components of the degradation system, ERAD, to get rid of hopelessly misfolded ones, even factors to physically expand the ER's capacity. So the UPR tries hard to alleviate the burden.
Yeah.
But its capacity isn't infinite. If the stress is too severe or goes on too long, the UPR might not be enough. Plus, Activating all those UPR genes uses up resources that could otherwise go to making our desired protein. And sometimes, if the stress just won't resolve, a chronic UPR can actually harm the cell or slow its growth.
Cell factory engineering mentioned actually manipulating the UPR, like overexpressing ATF4 or GADD34 in CHO cells, mammalian cells, could enhance production. So maybe not just letting the UPR run wild, but toning it.
Precisely. That's a more sophisticated approach.
Yeah.
Instead of just accepting the cell's natural response, maybe we can selectively boost the helpful parts of the UPR, the parts that improve folding capacity, without triggering the potentially detrimental aspects or resource drains. Carefully tuning the response could be much better than just letting it activate fully.
And secretion doesn't stop at the ER. Cell factory engineering mentions snares and SERT involved in vesicle transport later on. Boosting those helped in mammalian cells. Could that work in yeast, too?
It's highly likely, yes. Snares are the proteins that help vesicles fuse with membranes, which is essential for moving proteins through the Golgi and out of the cell. SERT is involved in lipid transport needed for those membranes.
So if transport is the bottleneck,
Then boosting the machinery involved, like snares or SERT-like proteins in yeast, could definitely increase the overall secretion rate. The basic mechanisms of vesicle transport are very conserved between yeast and mammals, so it's a very promising area for improving yeast cell factories too.
okay, so we've really mapped out the different ways making foreign proteins burdens these yeast cells. Now for the exciting part, how do we fix it? The mitigation strategies. The burden article listed several engineering targets.
Yes, it laid out a nice roadmap. Things like optimizing that gene dosage we talked about, carefully choosing promoter strength to control how strongly the gene is turned on, optimizing the codons in the gene sequence so the yeast ribosomes can read it more efficiently,
Count on optimization, right.
using better signal peptides for ER entry, making sure the growth media has everything the cell needs, and even redirecting metabolic pathways inside the cell to support protein synthesis.
And it seems like synthetic biology is providing the tools to actually do a lot of this, especially for dynamically regulating gene expression. Several sources hit on this burden. Cell factory engineering, control theory meets synthetic biology.
Absolutely. Synthetic biology gives us standardized genetic parts, promoters, terminators, regulators, and design principles to build complex circuits inside the cell. Dynamic regulation is key because the burden isn't static. It changes as the cells grow or conditions change.
Right.
So if we can build circuits that sense the cell's internal state, like how stressed it is, and automatically adjust protein production up or down, that offers a way to balance production with cell health much more effectively.
Control theory meets synthetic biology. Uses that great analogy, feedback control being like cruise control in a car. It's amazing seeing engineering principles applied to biology.
It really is. Control theory provides the math and framework for designing systems that maintain a desired output despite disturbances. We can build genetic negative feedback loops in yeast that say sense protein levels or stress indicators and automatically adjust gene expression to keep things in an optimal range.
Just like cruise control adjusts the throttle for hills.
Exactly. It could lead to much more stable and potentially higher overall yield.
That same source, control theory meets synthetic biology, had a figure showing the timeline of synthetic biology. How has the field's evolution helped tackle this burden problem?
Yeah, that figure shows how the field started with basic building blocks, simple switches, oscillators. But then researchers realized putting these into messy, noisy cells wasn't straightforward. They encountered burden, lack of robustness.
Reality bites.
Pretty much. So the field shifted towards designing more sophisticated circuits that could actually sense and respond to the cell's internal environment, including stress levels. Developing feedback and feed-forward control motifs was a direct response to needing more robust systems for things like protein production. We've moved from simple parts to trying to engineer resilient systems.
Figure 3 in that paper showed how negative feedback reduces sensitivity to uncertainties and disturbances. So in our yeast factories, what are those uncertainties and disturbances that feedback could help manage?
Good question. Uncertainties could be things like, well, slight variations in how strongly a promoter works, or how efficiently a ribosome translates the message, things inherent to the biology that make prediction hard. Disturbances might be external things. Maybe the nutrient levels fluctuate during a big fermentation run, or the temperature shifts slightly, or internal, like that plasmid copy number varying between cells we talked about.
Right.
Negative feedback can buffer against all of that. If plasmid copy number goes up unexpectedly, increasing protein production, a feedback loop could sense that rise and automatically dial back the gene's transcription rate, keeping the overall output more stable.
The article also mentioned feed-forward control as another strategy, maybe for dealing with that plasmid copy number variation. How is feed-forward different from feedback?
Feed-forward tries to anticipate a disturbances effect. If, based on a model that a certain change, like higher plasmid number, will cause a specific increase in output, you can proactively make an adjustment, like reducing transcription, before the output even changes,
Yeah.
aiming to cancel out the disturbance.
So it's predictive.
Yes, but it really relies on having an accurate model of the system, which is tough for biology.
Yeah.
Feedback is reactive, it measures the actual output, and corrects based on the error. It doesn't need as precise a predictive model.
Okay. And Figure 6 mentioned retroactivity, how connecting circuits together can mess up the upstream one, and using feedback to build insulation devices. Why is modularity important, and how does feedback help achieve it here?
Modularity is like the holy grail for synthetic biology. Being able to design independent genetic modules, a sensor, a logic gate, an output device, and know they'll work predictably when you plug them together.
Like biological Legos.
Ideally, yes. But
Mhm.
retroactivity breaks that. Connecting a downstream circuit, like our protein production module, can load the upstream circuit, maybe our sensor, changing its behavior. It stops being modular. Feedback can be used to design insulation devices, circuits that buffer the upstream module from the downstream load. They use feedback to ensure the upstream part maintains a stable output regardless of what it's connected to. This helps preserve that modular, predictable behavior, which is crucial for building complex systems like sophisticated cell factories.
The burden source also looked ahead, mentioning mathematical models and these dynamical regulation circuits as future strategies for better cell factories. How does those advanced approaches help?
Mathematical models let us simulate the incredibly complex network inside the cell. We can integrate data, test hypotheses in silico, predict bottlenecks, and guide our engineering efforts much more rationally than just trial and error.
It's a smarter design.
Exactly. And dynamical regulation circuits, especially those using feedback and feed-forward, allow for real-time adaptive control. The cell factory can adjust itself on the fly to changing conditions or internal states, maintaining high production while minimizing burden. It's about making them more robust and efficient.
And that thesis title, Feedback Regulated Cell Factories for Enhanced Therapeutic Protein Manufacturing, really hits the nail on the head. Can you give some specific examples of how feedback might actually be implemented to adjust production based on burden or the environment?
Sure, lots of possibilities. You can engineer a circuit that senses the level of unfolded proteins in the ER, maybe tapping into that UPR sensor, IR1. If unfolded proteins rise too high,
Yeah.
the feedback loop automatically reduces the production rates of your target protein, giving the ER time to catch up. Or you could sense key metabolites like ATP or specific amino acids if they get depleted.
Dial production back.
Right. Temporarily throttle production until resources recover. You could even link it to environmental sensors. If the temperature shifts during fermentation, maybe a feedback loop adjusts chaperone expression or protein production rate to compensate and maintain protein quality. The goal is dynamic, responsive control.
This has been a fantastic deep dive. It really shows the incredibly intricate balancing act inside these tiny cell factories. We've seen how basically everything from the extra DNA right down to folding the final protein impacts the cell and the final yield.
It truly is a complex interplay. Molecular biology, metabolism, engineering, all coming together.
And the strategies being developed are just amazing. Fine-tuning with non-coding RNAs, these sophisticated feedback systems, borrowing from engineering, it really shows the power of synthetic biology to tackle these challenges.
Yeah, the potential to make biomanufacturing more efficient and sustainable is huge. These interdisciplinary approaches are really driving progress.
And drawing inspiration from control theory, from engineering, it just highlights how interconnected science is becoming.
It absolutely does. Bringing different fields together is often where the biggest breakthroughs happen.
So for the learner and for all of you listening, we really hope this gave you a clearer picture of the complexities involved in making these vital medicines and enzymes using yeast. It's clear the journey to build better, more robust cell factories is ongoing, and while it promises some truly remarkable things in the future.
It's definitely an exciting field to watch and be part of.
Thank you.